I'm Dr. Brian Nickel, your cannabis expert for CannabisExpertMD.com. We're going to talk a little bit about a topic I've been meaning to address for some time, but with everything in the news, it looks like it's an appropriate time to bring it up. I'm going to talk about cannabis potency testing. Lots in the news recently about cannabis labs possibly inflating THC results. In fact, in Arkansas, there was even a lawsuit filed after patients found that the cannabis flower that they tested themselves measured significantly different from that which was reported on the label. Now, these plaintiffs say it's fraud. Is it? It did bring to light one of the issues with cannabis and cannabis testing, and that is that consumers need to understand how cannabis is tested and what these results actually mean. Cannabis plants are sampled. For every 10 pounds of flower produced, that is considered a batch. About 20 grams of cannabis is collected from each batch for testing. Different parts of the plant are selected specifically from this particular batch sample in the same plant. It is interesting to note that the very same plant will show different potencies based on where you take the samples from. Colas, these are the big buds found at the top of the flower. They are big because they are exposed to much more light. More light means more photosynthesis. Photosynthesis means that the plant can actually produce more mass. Now, the colas are also where the most trichomes are produced. Trichomes are the glandular part of the plant in which cannabinoids and terpenes are synthesized and concentrated. Now, there's also popcorn buds. People have heard of these. These are much smaller flowers off the cannabis plant. They tend to be produced on areas of the plant that don't receive as much light. They don't get as much light, so there's not as much photosynthesis going on. They can't grow as large. In addition to that, they have a tendency to show a lot fewer of the trichomes than the colas do, so we expect them to have lower cannabinoid and terpene potency. So several specimens from a single plant are combined together into a single sample. Again, 10 pounds of cannabis, it's a batch, results in a 20-gram sample obtained from different parts of the same plant. This is processed down to about a 1-gram sample that is used for testing. So right here, the tested sample, it's going to represent the average potency of the plant. It reflects hopefully, the potency of the entire 10-pound batch. The reported potency for a particular batch is nothing more than an estimate of that contained in all the flower available at the dispensary that you receive. It is not necessarily the amount that will be in the cannabis that's in front of you. Now, terpenes, they also vary by the part of the plant sampled. The top flowers, the colas, of course, receiving much more light. They produce more trichomes and hence more cannabinoids and terpenes than the flowers from the lower part of the plant. Even the types of terpenes that are produced will vary according to which part of the plant you sample. The colas up at the top, they have a tendency to produce terpenes like limonene, pinene, and geraniol. They have a tendency to attract pollinating insects to them. The lower flowers on the plant, they have a tendency to produce terpenes that discourage herbivores, such as beta-caryophyllene, that has a bitter taste. Now, beta-caryophyllene does discourage a lot of herbivores, but these bunnies here, they seem to enjoy cannabis a lot and have no problem with beta-caryophyllene. Again, terpene results are reported as an average of the plant examined that should represent what we expect to see in the entire batch. Again, we're looking at estimates here. Let's talk about THC potency. Now, how THC potency is reported can also cause a bit of confusion for people. Total THC, that is what most people look at on the flower that is obtained from dispensaries. Now, total THC, it's reported as a percentage of total weight. It measures THC in the particular specimen, all the THC present. There are a couple different forms of THC there. THCA, it is the dominant form you're going to find in the plant. This is the acidic form of THC. It is considered inactive. It's not the psychoactive portion of THC. THCA is converted to delta-9 THC through heating of it, a process which is called decarboxylation. THCA weighs more than delta-9 THC. When THC is decarboxylated, there is a removal of a carbon and 2 oxygen molecules as a CO2 molecule. Weight is lost. The amount of delta-9 THC produced from THCA will weigh only about 87% of the original amount. So delta-9 THC content will be 13% lower than that reported as THCA. The potency reported on the COA, it's based on that 1 gram of prepared specimen from the 20 grams of flour that supposedly represents a 10-pound batch. So again, the certificate of analysis, it is a best estimate of the potency of that flour and is supposed to reflect the potency of that entire 10-pound batch. Another issue when we're looking at potency is there can be a wide range of potency between samples. As an example here, we're looking at OG Kush oil. The 6 samples shown, they had a range of between 51 and 55% total THC. The average for this batch is 53%. Pretty good estimate based on what we see as far as the range goes. In the next example here, we looked at 6 samples of Stardog oil. Now, specimens from this sample range from 32% THC to 80% THC potency. It doesn't look like they got a very homogeneous mix in this particular oil. It does come out to an average, however, of 53% THC, just as we saw with the OG Kush oil. Now, the wide range and therefore what is considered a high standard deviation, that's the difference between what the actual average is and what the individual specimens measured, becomes quite high, showing the great variability between specimens. This really means that you have a very high probability of obtaining a cartridge with significantly higher or lower levels than were reported on the COA, even though the COA reports an average of 53%. So, take this all into consideration. There is a very high probability that the estimated potency of the flour that you purchased will vary slightly to significantly from that which is reported on a COA. Well, at this point, you're probably wondering, well, what good is laboratory testing at all then? Well, first of all, laboratory testing ensures that you get a safe product. Testing for pesticides, heavy metals. Heavy metals are very important to test for in cannabis. It's known as a scavenging plant for heavy metals. We'll pull them out of the soil and concentrate them into the plant. They are tested for coliform bacteria, for proper moisture content. Very high moisture levels promote the growth of mold, fungus, and mildew, all of which can have deleterious health effects. There was an illegal grow recently uncovered in California. It was being run by South America drug cartels. They were producing a lot of cannabis at this grow. When the cannabis flower they were producing was analyzed, it showed extremely high levels of many banned toxic pesticides present. Cannabis from the black market, it is untested, entirely unregulated. There have been some studies showing extremely high pesticide levels in some of these products, sometimes thousands of times the upper limit that is permitted in the legal industry. Now, getting back to the entourage effect, as I always seem to do in any topic that has to do with medical cannabis, this becomes important when you start considering, what am I going to do with the COA? Remember, the entourage effect really states that the exact potency of the cannabis and terpenes in a particular specimen really is not that important. It's really which of the cannabinoids and terpenes are present and then the relative ratios that are contained within that specimen. That's what really determines the effects that cannabis is expected to produce. The actual potency just reflects the dose that has to be administered. If one hit of a 20% THC flower has a certain effect, 2 hits of the exact same flower with a 10% THC concentration are going to produce the exact same effects. You just have to increase the dose a little bit. Knowing the cannabinoids and terpenes present, and the relative ratios, which are reflected in batch tests, again allows you to make a much more specific selection as far as cannabis so you can attain the results that you're trying to achieve. So reviewing the COA, this number one ensures that you have a safe product for use, and it will give you an idea about the expected effects of the cannabis being used. You have to remember, cannabis is a medication derived from a living organism, the cannabis plant. There's a lot of variability in what the plant produces. This is determined, of course, by the genetics of the particular species being grown, the amount of light exposure, ambient temperature, relative humidity, the water quality that's used to feed the plants, along with the fertilizers used, and even the atmospheric carbon dioxide content. Many cultivators add CO2 to the atmosphere to improve just not potency, but yield that they achieve on their crops. The complex nature of cannabis and laboratory testing, it makes the certificate of analysis interpretation much more complicated than just looking at the total THC percentage. Now that you have a handle on how cannabis is actually tested at a laboratory, the limitations of the testing procedures, the variability within a particular plant itself, the variability between specimens that can occur even within the same genetics, and that it is the presence of the cannabinoids and terpenes and the relative ratios that are important, you will be much better prepared going into the dispensaries. You can be able to ask for relevant information from the bud tender so you can optimize your use of this medication instead of walking into them and asking, what's your highest THC? For more information on all things medical cannabis, be sure to check us out at CannabisExpertMD.com. We're also available on YouTube, Instagram, Facebook, Twitter, Gab, and Relevant under Cannabis Expert MD.